All right, good morning, everybody. Happy Friday. We left off with some worms. I know you remember that. We've got a handful of worms that we want to go through a little bit of detail about some of the life cycle. And then we're going to transition and talk about some fungi. And then wrap it up just with the littlest bit about some lichen. And that will be today. Okay, so we did show with this, we ended last time with the platyhelminthes and just a quick, kind of couple of examples of some of them as we look at them. So the turbularians, the monogenia, these monogenia, basically it's a single host that they infect. And then we see trematodes, cestodes, and usually with that, there's multiple things that are participating in the life cycle, at least two hosts, sometimes a handful more. So these on the bottom are the ones, the trematodes and cestodes that we think of as being parasitic. So flatworms, tapeworms, flukes, they're all flatworms, flukes and tapeworms, respectively. So we want to start off thinking about them, getting a little further detail. It could be, although it's not, this is Taenia saginata, the beef tapeworm that we can see here. This next one is Taenia solium, the pork tapeworm. There are lots of similarities. There are some differences as well. And as we look up here at the head, if you will, the scolex, with Taenia solium, they have some ornamentation, some hooks up here on the scolex, whereas Taenia saginata does not. It's just got some suckers. So they also, as we say, beef and pork tapeworm, we get infected dependent upon the type of meat you might be consuming that's not cooked thoroughly. The first one, this Epistolorchis viverni, these guys, you can see the oral sucker up at the top. We can see some details within them. This, it's like the image we were looking at with the flukes and the tapeworms, right? So these guys are flukes, a flatworm. This is a liver fluke. And then next door is our tapeworm, although we're only seeing the very tip of these guys, of this one. So there are all sorts of potentials. And one thing, as we're looking at the Epistolorchis, these guys, well, same with the tapeworms, but you can't see it in this image, are hermaphroditic. So they've got both male and female reproductive structures within the worm, and fertilization occurs, releasing numerous, potentially hundreds of thousands or millions of eggs could be generated from some of these parasites. Probably not these guys right here, but definitely they hold potential to generate massive amounts of eggs from a single worm. All right. So this next one, these guys are a type of fluke as well, but they're the littlest bit different, and they're not hermaphroditic. These guys have separate sexes, and this image, little image below, shows the male and the female together. They are truly an exception when we think about the flatworms. These are the ones that are separate sexed. Everybody else, when we're thinking these parasitic ones, they're hermaphroditic, all right? We'll move in and think about nematodes, the roundworms, and those ones are separate sexed, or what we call dioecious, so individual worms, one's male, one's female, all right? So the schistosomes, the life cycle of these are really pretty complex. pretty impressive. It requires us to come in contact with some water. We need to be in an aquatic environment. So you also need to be in the right place at the right time, all right? Fortunately for us here in, well, here in Idaho for sure, but as far as I know, you don't get any sort of schistosomiasis in the United States, period. If you get it, you've traveled abroad. And almost always we think about some excursion to Africa that sets your stage for contracting this. There are all sorts of opportunities to come in contact. Going into some water sets the stage, all right? There are other locations as well, and we do see some in South America, but it's definitely more of an issue when we look in areas like that. We will think a little bit. There are some potentials for us to become infected with some schistosomes, but it's not these guys, all right? It doesn't progress and the parasite's not able to complete its life cycle. What we are infected with is a parasite that would be typically targeting some sort of waterfowl, a bird, a duck or something like that. But we get into the water and it ends up getting into us instead, and it stays localized in our skin. We call that stuff swimmer's itch, and it is something that is pretty stinking common. You can see it in all sorts of different waterways here in southeastern Idaho. Rigby Lake's got them, Jensen's Grove has it. There are definitely other places where you might get in and find yourself potentially with some of this. It is a self-limited infection, so basically our body's going to kill these things off pretty quickly. You've got a few days of feeling some discomfort as the parasite's trying to live under your skin, and we destroy it. And that makes me feel a little creeped out, all right? So we'll go through this as we think about the ones that truly cause human infection and not the ones that we're thinking about that are just get under your skin and creep you out a little bit, all right? So there's all sorts of details as we move through here and look at this life cycle, all right? Starting down below with our number one, there are a handful of different species of schistosomes that do cause actual disease in humans. With that said, we can differentiate them a little bit, and you can't see great details with these eggs, but that's what we're looking at here. The eggs of these are different. Some have some spines, some little spikes on them at the end or on the side. Another one just kind of rounded, no spike. You can see a spike right here, nice and good. So with that, there are also differences in the body where we might find them. Most of them we think about being in the GI tract, eggs being released in the feces. One of them, where is it at? Hematobium, which is this egg right here. Hematobium you find in the veins going into the bladder, and the eggs are generated by these worms and released into the bladder. So the eggs with these spikes cause damage. There's potentially some damage when we think within the intestine, but it's more with these with their lateral spine that inside of the bladder causes damage, causes abrasions, lends to rapid cell proliferation and potential bladder cancer, right? This also would be something that we think about with a prolonged infection, something that you've had maybe for years, lending to compounded effects, right? So it depends on the circumstances, all right? So either way, wherever these guys are out, the eggs get released from us, either urinated or defecated out, if they happen to make it to water, which is what needs to happen, these eggs will hatch. They release out a stage of the parasite that looks really like a protus. It's ciliated, it swims around. It's not a protus, it's a juvenile worm, all right? This thing swims around and it finds a snail, an aquatic snail, all right? It penetrates into the snail, and as it's penetrating in and moving into the tissues of the snail, it sheds that ciliated epithelium and it becomes another structure inside of the snail that's a reproductive step, all right? So it generates sporocysts, and really each of these steps kind of in the snail harbor the next stage, which is really pretty crazy. So the Myristidia converts into this sporocyst, and inside of the sporocyst are the cercaria, and the cercaria are the next step that's going to be what gets us, all right? So the cercaria, after rounds of, time, not rounds, but time for development within the snail and feeding, the cercaria are released. They penetrate out of the snail. They've got some enzymes up in their head region that allows them to bore out of the snail tissues. And then they get into the water. They swim around in the water. They can survive for a couple of days in the water, swimming around with the hopes that some mammal is going to come around and they can penetrate in. That's really probably what's happening. And really, it's us with these species that end up being the ones that truly want to be targeted, all right? With the swimmer's itch, we get into the water, but we're the wrong host. We're not a duck. And the parasite penetrates in, just like these guys are, but instead of being able to migrate through the tissues, it stays localized. That's not the case with them. So they penetrate into our skin. They ultimately migrate through our circulation. In this case, this little schistosomule, as it gets in, we convert, it gets into the circulation, and from there migrates either into the hepatic portal veins that are going to be dumping eggs into the intestine, or into the bladder plexus where they're going to be dumping eggs into the bladder. Within the liver, before we make it to our final spot where we want to, build our little house and start living, we're going to find one another in the liver, all right? So these worms, as they get in and develop into the adult stage, to the fluke, they sense one another, and it's within the liver where they mingle and find a significant other. They pair up and basically stay locked in love, and will constantly be copulating, and the female releasing copious amounts of eggs. Really pretty crazy. They're called schistosomes because the male, his part of his body is split open. They call it the gynecophoral groove or canal. It's basically an area that the female will slither into, and then he sandwiches around, and that's it. Now they've paired up, and then they migrate to wherever, right? The intestines or the bladder.
She's completely inside him?
Not completely. So you can see these guys right here, they're sort of slithered together, right? So, and I don't know if we've got another image of them or not, no, but not completely inside, right? Just kind of sandwiched together, all right? And there are images, and if I would have been on my game, I would have brought my picture in here. I will try to remember and bring it for Monday. There are pictures that show these things, and they look so disturbing. They're massive. It looks like these gigantic monsters, but they're living in your veins. They're not gigantic, right? These things are virtually microscopic. They are super, super small. right here, they also look kind of terrifying, kind of monster-like, but they are super small, right? So that also sets the stage for, some of their potentials to truly cause these ill effects and infection by maybe, circumventing some of our responses. And there's all sorts of different details. They're all of these worms. I'm like, I could talk all day. You know that from last time. There are definitely circumstances once you get infected with these, and you're going to probably have a good handful of these worms within your system paired up, all fine and good, because when you first got infected, it was probably numerous cercaria that penetrated into you in the first place, right? Once we finally get to our specific spot and we've got these adults pumping out these eggs, there is a bit of an immune response that gets stimulated, but it doesn't do anything to the worms that we've already got going on. It gives us a little bit of protection from other cercaria that want to penetrate in and cause infection. These guys are like, we're already here. You can't move in, right? And so it causes our response to basically mount a defense. When other new cercaria try to move into our bodies, we try to kill them all off, right? So it's a mechanism that helps us not get a super infection where you've got just constant bombardment of parasites, but it also allows those that took up residence first to keep living, right? And for a parasite, that's a good thing, right? For us, that's not a good deal, right? But for the parasite, it definitely is, all right? So really pretty cool stuff. The snails are specific to lend to these guys that infect humans, right? In order for that life cycle to persist. We don't see these types of snails around here. We don't see these parasites around here. The swimmer's itch utilizes a completely different snail. And again, the host they want to be getting into is not us, it's a bird, right? And so it, again, stays localized. Okay, this next one for our tapeworm, we're really slamming both of them together, the beef and pork tapeworm, because there is a lot of similarities, right? There are a few morphological differences. There are definitely some differences as far as how the disease might progress. But overall, pretty similar, all right? So our life cycle here, the CDC has all sorts of awesome stuff, all sorts of different images with parasitic infections on life cycles. They've got other stuff with bacteria as well. But some of the parasite life cycles are really pretty impressive. So if you are interested, you definitely can search out some of this stuff and find yourself all of a sudden a couple hours later, you're like, holy cow, why? All right. So as we look at this, we've got our pigs and our cows that set the stage for the solium and saginata respectively. The idea with this, the livestock ingests the egg. The egg must be released in the feces of something like us, right? We are the one that harbors the adult stage of the worm. There are potentials for some other mammals that might serve this purpose, but typically thinking about these guys, we are the ones that really are harboring the adult stage of the worm, all right? So we defecate these eggs out. As we think of, first world nations, most of us aren't just walking outside and pooping in the rose bushes, right? Probably not, right? I did hear about some lady that, she'd go out jogging and halfway through her jog, she'd always end up having to go to the bathroom, and there was this one yard she'd stop in. Like every stinking day, right? And the owner is finally just like, what's going on here? He keeps crapping in my lawn, and tried to, set up some sort of camera to catch her and whatever, with her pants down. So you never know, right? But I would like to think most of us don't just randomly poop in people's yards, right? Or out in the pasture, because most of us also don't have cows or pigs out in our yard either. So if these guys happen to be able to consume some of this stuff, and there's no reason they wouldn't, all right? I'm not going to go into details about that, but they definitely, they're like, that's delicious. I don't know why, but they would, all right? As that gets consumed, the eggs that could be within there, there are also potentials for individual segments. We call those proglottids, one little piece or fragment, sometimes a couple pieces that have the mature eggs within it. They may be defecated out, the eggs may rupture, or the proglottid may rupture, releasing the eggs, and that's defecated out. Either way, it gets ingested, the egg hatches as it moves into the digestive system of the animal. It stimulates that egg to hatch. It bores through the intestine of the animal and makes its way into the musculature. As it gets into the muscles of either animal, it basically forms a cyst, and it lays kind of in wait, right? That animal in turn gets harvested, butchered up, and we eat it, right? If that meat is not thoroughly cooked, the larval stage is still viable within that, all right? And then it gets consumed. Then that larval stage, as it moves through our stomach acid and makes it into the intestines, the stomach acid stimulates it to say, hey, we're in business here, right? And now we get the adult stage of the worm growing within our intestine. Dependent upon who it is, like I said a second ago, the Saginata, no hooks, the Solium has hooks, both of them growing in our intestines, all right? They get to the adult stage. Some of these, I mean, it kind of just depends on the circumstances. A couple of meters, maybe up to 5 meters in length that they can grow. There are other instances in like some other countries, beef tapeworms potentially reaching 20 meters in length. Typically around here, about 5 is all you're gonna get, right? You want a bigger one, you gotta travel abroad, right? Why? I don't know, right? I would think maybe some subspecies variation potentially, but I don't know, all right? So all of this, we ingest it, it gets in there, we start producing the eggs that get released out. It's the same idea. These worms are hermaphroditic. It only takes one of those cysts, the larval stages, to be ingested, and now you've got an adult worm, all right? Dependent upon how many were ingested, maybe now you've got worms, right? Not just one. One detail with this, the beef tapeworm, all you get is the adult stage of the worm, all right? You ingest the poorly cooked meat, you get the worm in your intestine, and that's it. The pork tapeworm is different, right? You can potentially, through poor hygiene, you can potentially ingest an egg, just like the pig is doing, all right? You can ingest an egg, and then it's the same idea as if you are the pig. That egg hatches in your intestine, bores through, gets into your musculature, and forms a cyst. It redirects the blood supply and nourishes this larval stage. But for us, technically that point of it is a dead end because you're probably not going to be consumed, right? So those larval stages will die with you. It also depends on where they might decide to do that for the potential for some issues, definitely, all right? So in the case of the pork tapeworm, we say that we have the opportunity to serve as both the intermediate, like the pig, or the definitive host with the adult stage of the worm in your intestine, all right? And the beef tapeworm, we're only a definitive host with the adult stage found within our body. And the cow or pig, these guys are intermediary hosts, even though we say it's a beef tapeworm. it's truly infecting us because that's where we see the adult stage, all right? You can see over here, there are some differences as far as how the proglottids appear. You can definitely, as you're looking at them, and we can't really see this stuff now, but you can see, just like this one, inside each of the proglottids, there's all sorts of reproductive structure, right? An ovary, numerous testes, and then a bunch of other cells that we call vitelline cells or vitellaria that basically are going to coalesce and form around that egg as it's being generated. The vitelline cells provide the nourishment to that egg, kind of like yolk, basically kind of the same idea as yolk. That all over in this fluke, right? The bulk of the fluke is reproductive structure, right? And it's no different when we talk about the tapeworms. Each of these proglottids is basically just a massive amount of reproductive tissue, right? Ovary, testes, and all of the other stuff that's needed, all right? With that said, the flukes and our cestodes over here, our tapeworms, they've got suckers, they've got oral suckers on the other one, but they're not really aiding in digestion. These guys, it's just to stick, it's a hold fast, it's a suction cup is all. They absorb their nutrients right through their skin. So the whole body of this worm is pulling in nutrients just through its tegument, right? Through diffusion. And the bulk of what it's pulling in is glucose. Simply moves right in and it metabolizes it, all right? And all of these guys that are parasitic, as they're pulling in nutrients, they don't really care. It's like you took them to the buffet and all they want is the dessert and it's just fine, right? We're just going to eat that and throw the rest away, right? They're very wasteful. They eat stuff and only partially eat it and discard the rest as waste. Doesn't matter. I'm constantly bathed in nutrients. So why would I finish all of this? I'm going to take a bite and move on and take another bite, right? And just taste all of it, but not fully consume it. And some of that sets the stage for ill effects, right? This next one is impressive, disturbing, and something that we are on the cusp of having this parasite eradicated, or so we think, all right? There are definitely issues, and little Fido down here is perfect because we feel like we've about nailed these guys, and they don't really cause huge issues in humans anymore. But we do recognize that stuff like dogs are also serving as hosts for these worms. So we thought we had a pretty good handle on it, and we're like, wait a minute, we haven't truly eradicated this parasite. It's living in other animals now, right? That's a problem, all right? So it opens up the idea that we've got to do more treatment, right? It's more than just thinking about people, it's others as well, right? That sets the stage for some of these issues. So the guinea worm is something that is really pretty impressive. It is a roundworm or a nematode. And you can see here the female and the male, and the female can be really quite large, up to about 1 meter. The male, not so much, all right? The female, and it's kind of pinworm-like, with the male copulating and dying shortly after, and then the female basically is just pumping out mass quantities of eggs. This is a kind of disturbing idea as we think about it. It's not something that just happens overnight, all right? This is an infection that takes at least probably a year, maybe even longer, before you ever recognize that you've got an issue, all right, before the ulceration happens. And so we're looking at the back, probably the heel of a person, where it has ulcerated and the worm is protruding out. This is a matchstick. They grab the worm and they twist it slowly onto the stick. And then every day, just a little twist, because these things are almost a meter long. You can't just pull it out, right? You have to slowly twist it for a long time, a week or longer. It's going to take to just slowly pull this sucker out, which is disturbing in and of itself. If you break it off, that's a bigger problem, all right? You want to be very careful, all right? What is it? The caduceus, the medical insignia with the staff and the serpents, they speculate it's really this. That's what it signifies. We will come to us. We'll remove your worms, right? Which are snakes, right? Serpents, but truly parasites. Awesome. So we get infected from these crazy little copepods, an aquatic invertebrate, some sort of whatever it's going to develop into. We accidentally ingest it as we drink it down. There are other potentials where it could be a parentenic host, something else. The fish eats the copepod, or a frog eats the copepod. And then we eat either of these straight up. We just eat it raw, all right? If you're going to eat a frog, you ought to cook it, right? You probably ought to cook your fish too, but steak and sushi is so tasty. Who knows? So we can through undercooked things, but typically we think about water, all right? We drink that copepod. It in turn releases the larva that's within the copepod, the juvenile stage, and then we get to the adult stage within the intestine. There's mating. The female makes her way through the circulation into the skin, and this is where it takes a long time. All right. After all of this has occurred and she starts her migration and maturation, it's almost a year, maybe even a little bit longer than a year before she's finally ready and a meter long, finally big enough to start pumping out all of those eggs. So the ulceration happens. You know that sucker hurts. What are you going to do? Put it in the water, right? You want to soak it, and that's what the worm wants you to do, all right? As you put your foot in there, she's like, sweet, and starts just spewing out eggs, releasing the eggs into the water, which in turn will hatch, or releasing the larva, excuse me, into the water. She's giving birth to her live young. The larvae are released, and then the copepod sees these little guys and eats them, right? And the cycle continues. It used to be something that was really a pretty big deal when we thought about some parasitic infection. And then we start thinking some details about this life cycle in ways that we can maybe through thinking about water purification, the way that you're getting your water in the first place, filtration mechanisms. There are definitely areas where you've got like these big step wells where you just walk down and your feet are probably going into the water as well. And dependent upon the water level, you scoop your water up and you're back out of there. Others where it's just a big watering hole and you're drinking out of it, and so are a bunch of other things, right? All of these, you know, as I'm describing it, as we sit here, we're like, that's crazy. Why would you do that, right? It depends on where you're at, right? It truly does. And that sets the stage for this, all right? So it was something that was really a huge issue, and we got to the point that it's only a handful of cases, some like a dozen or maybe 20, something like that. Treat them, and we're right near eradication. We're not seeing human infection. But now we're seeing lots of veterinary issues with dogs. They're still going down to the water too, right? And in turn, they've got ulceration, and it's the same issue with the worm protruding out, and the dog goes to the water and the larvae are released, and it keeps it persisting within the population. Or within the environment. Crazy. Okay, we're going to shift from that and think a little bit about some fungi, right? Again, I could have spent all day thinking worms. Fungi, there are all sorts of details. And really, when we think about it, it's all over the place, right? I did not look at all of your environmental plates. I did take a peek in the incubator and see some stuff, but I didn't look at, the 100 plates or more than 100 plates that were in there. But my gut tells me some of you guys had some environmental stuff that probably had some mold growing on it. You probably saw some fuzz. And you could have sampled all sorts of different things, and highly likely you're going to find some of that stuff. All right. You also might find some of it in like the back of your refrigerator, and you're like, what the heck's in this Tupperware, right? And it's not what you thought it was, right? That always gets me a little bit disturbed. I'm like, holy cow, I could have eaten that, right? So this is kind of thinking like a microscopic view. It's still a cartoon image, but like microscopically what we would see. So with our molds, if we were to look at them under a microscope, they may look one of two ways. This first one has lots of little divisions in between all of the dots or the nuclei. The separation we call septa. These are septate hyphae. The hyphae are the strands, right? The filaments. A whole collection of hyphae, the fuzz, the mold on the side of an orange, or in your, you open up your loaf of bread and you see some mold in there. All that whole big mass of mold that you see, we call that mycelium. It's a collection of hyphae, right? That's mycelium. So here we're just looking at a few threads, the hyphae, all right? The divisions we call septate, and then the cenocytic or non-septate, it's all a continuum, still separate nuclei in there, multinucleated, if you will, but you don't see individual divisions, right? Next door, the third ones, these are the pseudohyphae. These are yeast cells, not molds, all right? And some yeasts have the potential to produce structures that resemble the hyphae, but it's not true hyphae. It's not real filamentous forms. It's just part of the process of reproduction, and some of it is also potential to lend to virulence, to disease causing properties. So Candida albicans is a yeast that is one that can produce these pseudohyphae, and it's a pathogen in us, right? And we'll see some extra stuff with that later. So there's all sorts of details with fungus that I think is pretty cool. One, not histoplasma, but one that's similar to histoplasma was the focus of my graduate studies. It was called Blastomyces dermatitidis, kind of cousin-like, if you will, as far as what's happening, all right? There are a handful of parasites, or fungi, I should say, that end up kind of being parasitic within us as they get into us. There are a few that we say are dimorphic fungi, all right? And histoplasma, the blastomyces, and coccidioides are the three main ones that we think of as being dimorphic. What we mean by that is this fungus has the potential, and probably the majority of the time, it's going to live as a mold. And it's got hyphae, it spreads out, it's breaking down stuff like rich organic matter, it likes bat feces and bird droppings, high nitrogen content is a situation for this fungus to really kind of thrive in, all right? So it grows in these types of habitats. So as it says here, soil exposed to the feces. And the next bit is where it sets the stage for the dimorphism. So at regular room temperature or whatever, 70 degrees, it grows as a mold. The hyphae, it produces its reproductive structures called conidia. They're going to burst open and release these spores. that will settle back down into the environment and will continue to grow as a mold. All fine and good, all right? But if it happens that we come upon this stuff as those spores are being released and we inhale them, now all of the sudden, these things that would have just settled back down in the back crap get into your lungs. And those spores don't go into this hyphae stage, this mold stage, they convert into a yeast phase. The dimorphism is the capacity to grow as a mold and as a yeast, and typically it's a temperature dependency. You go from 70 degrees up to 98 degrees, right? And as soon as you ramp that temperature up, plus a nice moist environment inside of the lungs is also pretty good, right? All of a sudden, these yeast cells generate, they start replicating by budding, they proliferate, they potentially spread. You can see them within the lymphatics, the circulation. There's potential for skin manifestations, central nervous system involvement. We'll mention some of the dimorphic fungi in repeated chapters at the end of the semester, because there's a possibility to see disease process throughout the body, all right? Awesome and disturbing, all right? These guys kind of Midwest, Ohio, Mississippi river valleys, both of which we also would say is where blastomyces is endemic as well, all right? We don't see them out here, all right? Coccidioides is one that likes a drier climate, more of a desert-like climate. Southern Utah, Arizona, New Mexico, Southern California for sure. It's been called valley fever for the San Joaquin Valley. It's in those types of habitats. And as we see climate change, it's definitely something that should be on our radar, right? It's not, that doesn't live that far away from here, all right? And the potential for it to possibly thrive in this habitat, once it gets here, I think it's going to be happy, all right? So similar things can kind of occur with all three of those. As they get into your lungs, replicate, and then ultimately disseminate to other body sites. Kind of crazy. So dimorphic fungi, there's a few, and they definitely set the stage for some serious disease. There are other considerations. I'm not going to get bogged down with that stuff, but it definitely depends on your circumstances. There are all sorts of types of fungi, and we think of different types of molds, but there's other things. Yeast fit within there. You could think about something like a mushroom. That's also a type of fungi. And we categorize these guys based off of some of these specific features. We think about, and it is really pretty crazy and confusing to think about some of these details. Lots of the fungi, we can see different stages or different phases where there is a sexual phase and an asexual phase. There are some where we only know an asexual phase. That doesn't mean they don't have a sexual phase. We just haven't figured it out yet, all right? It's highly likely that they've got both, but we're not sure, all right? And so they get grouped up based off of some of that stuff and how it ultimately functions. So here we can see some of the, here it says mycelia, but really it's the hyphae because we're only showing you two, right? But we've got two mycelia right here. These going through their asexual replication in the top bit and sexual replication in the bottom bit, all right? And it kind of just depends, again, on the type of fungi as we move through here and the types of structures we see. There is a huge amount of similarity. It's just some subtle deviations with like some of the spore forming structures that you might see or the fruiting bodies, whatever, right? So up here in the asexual phase, the mycelia can go through their asexual replication through mitosis and generate more. They can produce these spores, which in turn germinate, and it's the same as up above. We see the hyphae or the mycelia, all right? But in this case, as we look down below, we've got, and it's hard to kind of see, but different colors in here. I mean, if you looked at it under the microscope, you're not going to know, all right? You can't tell, and you can't see this one's strand, this one hyphae, and say, oh, that's female, and this one's male. We don't call them male and female, but it's kind of like that, all right? We call them opposite mating types, plus and minus, all right? Who's the male? Who's the female? We don't give a crap about that. We're not talking about that. There's no sperm and eggs being generated here, all right? It's just opposite mating types. And that's why they've tried to show the nuclei as red or green dots up in there. If that's green, I don't know, right? It's a different color, all right? When I get close to the monitor, I think that's maybe yellow. I think now maybe I'm colorblind. What's going on here? So the opposite mating types basically come together, and you can see here positive and negative, and we fuse. They form a zygosporangium, and in this case, it's not really like the fusion of egg and sperm. We get two nuclei together, all right? We do say, diploid at this point, because there's two copies, right? Diploid nuclei. This zygote that's diploid goes through meiosis. As it germinates, it produces the fruiting bodies that are haploid, which burst open. It's the fuzz on the side of your orange, right? Full of all of these little spores. It bursts open, releasing the spores. And in turn, dependent upon what's going on, these spores can just go through asexual division, all fine and good. And maybe all of the sudden, one of the hyphae is next to another hyphae, and they are opposite types, and they fuse, right? So it can be numerous asexuals and then a few sexual divisions. If it just happens that the two threads are side by side and they're opposite, then this can occur, right? And as it does, they produce the sporangia, and the cycle just keeps going, right? So same stuff, just dependent upon kind of placement or proximity to one another as far as what's happening, right? So here we can see some bread, right? With these guys, the bread mold, and zooming in on them, the sporangia, really the dark colors in this fuzz are those fruiting bodies, or the spore forming, or the structures holding the spores. And that's what we're looking at right here in the microscopic image. So the hyphae and the spores, right? Here's just another type of them with the types of spores, the ascus and ascospores, and some microscopic images. So a couple of different types of fungi within there. Aspergillus, a handful of different types of Aspergillus fungi. We oftentimes call these guys black molds or toxic fungi. And it sets the stage definitely for some issues. There are potentials for it to get into your lungs and grow as a big mycelial mass. Call out a fungus ball. Makes me think of a cat trying to hack something up. But it's a fungus ball, right? It's not a hairball. Kind of crazy. And there's also potentials for some cancer-causing issues, liver cancer and stuff, with some of the toxins that some of the Aspergillus might be generating. Here's this image with some yeast. This is Candida albicans, and oval, sort of thick cell-walled structures. I think I'm basically out of time here. We're so close to the end, but we'll start with just the littlest bit of these guys on Monday, and then we will move into some virus. I hope you guys all have a good weekend. Farewell.